Assalamualaikum warahmatullahi wabarakatuh. My name is Fazilatun binti Abdul Malek. In this video, I will present about my poster that I've been reviewed from a paper, and the title is Analysis of the Genetic Diversity of Influenza A Viruses Using Next Generation DNA Sequencing.
I will start with the abstract. Influenza viruses exist as a large group of closely related viral genome, also called quasi-species. The composition of the influenza viral quasi-species can be determined by an accurate and sensitive sequencing technique and data analysis pipeline. In this experiment, two benchtop next-generation sequencers for whole genome influenza A quasi-species analysis were compared using Illumina MiSeq sequencing biosynthesis and the ion torrent PGM semiconductor sequencing. Based on the result, Illumina MiSeq sequencing rates were 1.5 more accurate than those of the ion torrent PGM. This approach underlined the power and limitations of two commonly used next-generation sequencers for the analysis of influenza A virus gene diversity. It can be concluded that Illumina MiSeq platform is better suited for detecting variant sequences. We move on to the introduction. Viruses outnumber all other known life forms on Earth. Furthermore, viruses in general and RNA viruses have a huge genetic diversity, which is a driving force of their evolutionary success. Viral genomic diversity is well captured in quasi-species. Human influenza viruses represent a prototypical example of rapid virus evolution facilitated by error-prone genome replication combined with the selection pressure imposed by host human response. This situation favored the emergence of fit mutant viruses that escape the herd immunity induced by infection with parental viruses or by vaccination. Influenza is an acute and highly contagious viral disease of the respiratory tracts in humans. It is caused by influenza A and B viruses and occasionally by influenza C virus. The identification of extant genetic variation in viral quasi-species under a given condition requires a highly accurate sequencing method that does not rely on molecular cloning or phenotypic selection methods such as plaque generation. Next-generation sequencing seems to fulfill this requirement. NGS enables sequencing multiple gigabases of DNA in a single run depends on the sequencing instrument. Consequently, because of the influenza genome consists of 13,000 ribonucleotides, it is straightforward to sequence it in a high coverage. There are two main objectives in these studies. The first one is to compare the performance of two high-throughput sequencing instruments, which is the Illumina MiSeq sequencing and the ion torrent PGM semiconductor sequencing. And the other one is to determine the complexity of an influenza A virus quasi-species, which means to count the number of nucleotide variants present in the swarms of genomes of the virus. Next is the methodology. This experiment was started with the sample collection of influenza A viruses, followed by the preparation of cell line, generation and production of plasmid with tracing mutation, generation of recombinant PR8 and PR8 mutated viruses, plaque assay, ion torrent PGM chip sequence determination, Illumina MiSeq sequencing determination, RT-PCR, RNA isolation, analysis of sequencing data, statistical analysis, and lastly are sequencing data. Continue with the result and discussion. The experiment was done by comparing the accuracy and sensitivity of the sequencer using plasmid DNA. The plasmid DNA were generated with two tracer mutations in order to determine the sensitivity of the sequencer for picking out the occurrence of the introduced SNPs. The plasmid chosen for analysis are the ones that allow to generate PR8 virus with or without the introduced tracer mutation. To evaluate the accuracies of both sequencers, the process read were mapped to the plasmid reference sequencer. The percentage of unmet bases was lower in the Illumina MiSeq other than the ion torrent PGM. This is due to the lower quality of the ion torrent PGM sequencing reads, which reflects the intrinsic sequencing error that leads to reduced alignment and higher number of unmet bases. Illumina MiSeq sequencing provides higher quality and trimming read length is greater than the ion torrent. Next, the determination of the number of variants at each nucleotide position in the virus-derived sequences, which will reflect the quasi-species diversity of in vitro-grown PR8 and PR8 mutated virus. The number and types of nucleotide variants that were retained after applying the variant filter are presented in the figure 1. The frequencies of the sequencing variant detected by both sequences reveal 90 mutations for wild-type PR8 and 29 SNPs for PR8 mutated. Nearly all SNP were detected with high average pH rate score on the Illumina MiSeq and were thus more reliable than on the ion torrent PGM. Finally, the conclusion. The study underlines the power and limitation of two commonly used next-generation sequencers for the analysis of influenza gene diversity. They also propose an in silico pipeline for selecting high-quality reads obtained by the NGS platforms. This pipeline is also widely applicable. Due to the lower total error rate and high sequencing quality of the read, it is concluded that the Illumina MiSeq platform is more suitable than the ion torrent PGM for detecting variant sequences, whereas the ion torrent platform has a shorter turnaround time. In addition, we found that detection limit for reliable recognition of variant in the viral genome require frequency of 0.5 or higher.